data for drug repositioning, a case study in glioblastoma. Some background of that research, this was well, still is an ongoing collaborative project between the Davis Center in Core and two neurosurgeons of the Curdin Hospital of Winterthur. Those two neurosurgeons developed an innovative combined regimen of 9 repurposed non-oncological drugs combined with metronomic temolol for the treatment of glioblastoma, which is the most malignant brain tumor. And our overarching research goal is to predict new possible drug candidates for repositioning by harvesting multiple different data sources and employing machine learning or deep learning techniques. This part of the research started as a master thesis with my main research question: how can unstructured data be used to identify new drug repositioning candidates, and how can they complement databases? Some introductions into the whole topic of drug development and drug repositioning. Firstly, the development of and discovering of new drugs is risky, costly, and takes a lot of time. The success rate of a newly developed drug to be clinically approved and to reach product markets is below 10%, which is not much. And as a consequence of this, many newly developed medical compounds end up abandoned or just lost to time. What is drug repositioning, or also known as repurposing? It describes the process of identifying and developing new uses for already existing drugs or known active compounds, in which we try to, of course, discover new use cases of known drugs by harvesting knowledge of previous research, and which is in general highly beneficial commercially and also for patients, especially for patients with rare diseases, because there is not much of a commercial interest often there to develop a new drug. Some basic knowledge which we can use for this drug repurposing process is that the similarity of drugs can be determined by chemical structure, overlap of molecular target structures and side effects, and the similarity of diseases can be concluded through ontologies or shared treatment profiles. Why text data? A huge amount of exclusive medical knowledge is hidden in various types of unstructured text data, such as clinical reports, scientific research documents, or journals. Especially clinical reports provide new knowledge that is not often recorded in standardized type databases or summarized medical literature. For example, on new occurring side effects or individual drug-drug interactions. Yeah, and also there's potential that there is buried information or knowledge on abandoned medical compounds which could be rescued. On that topic, there are some historic rescues of drugs. One famous example is zidovudine, which was developed in the 60s, and then later in the 80s was discovered to have a use for the treatment of longitudinal vertical HIV transmission. So there could be another drug there, it could be another alternative use to do it. The goal of this research was to firstly explore, test, and evaluate different approaches or methods to predict new drug repositioning candidates using NLP on open and publicly available unstructured data, and in which we developed or tested 2 different workflows to identify drug repositioning candidates for possible therapeutic use on our case study on glioblastoma. And for that, we combined mostly pre-trained biometrical naked entity models, which we could use to identify possible relationships between those biometrical entities. What tools did we use? Pretty simple. We used the SciSpacey library, which is available on GitHub. It provides fast, easy to use, and robust biomedical and EA models. And yeah, it offers, what's most important part, it offers 4 different specialized biomedical and EA models which enable a more wide subject scope of application. And it also provides an entity linker, which can be used to match results with controlled vocabulary, the unified medical language system. What text data did we use? We extracted, based on that point on the similarities of diseases, we extracted all clinical trials from clinicaltrials.gov, that is a big public clinical database, where we extracted every study on the topic of ectodermal tumors, which is a broader term for glioblastoma. We supplemented that with 3,059 abstracts from PubMed, and we had a resulting data set of 10,000 documents. Our first method, we just used a simple co-occurrence analysis based on the guilt by association principle. We used this co-occurrence of those NER-extracted biomedical entities to determine the most similar document pairs or the most similar clinical studies which were there, where we only focused on different variations of entities. With those most similar document pairs, we extracted and associated new drug disease pairs. And with our second method, we used the open discovery model of chains of associations, which is like by building associations chains to identify new possible transitively relations for new repurposing candidates. And in this method, we use public available databases to extract first A-B relations in the discovery process, and then we use those B terms as mostly search terms for text data to identify some hit documents where we could determine some preset entity type C for a transitive relation for our first selected A term. Those were the variations we tested on our method of our methods. On our first methods, we had on our first method variation, we just looked at the overall general biomedical entities. In the second variation, we used like overlapping terms of gene ontologies, sequence ontologies, gene products, or from 2 different models. With our third variation, we looked at diseases and side effects, because most named entity models can't really differentiate between symptoms, side effects, or diseases. So we had to work with the disease term from our third SwissText model, and where we used like the more specific bio-NLP model to like filter out unwanted entities, which weren't really could be a side effect. For example, cancer couldn't be a side effect of cancer, for example. Other things we use for a more open approach, we use the cells, and cell lines, and cell types for overlapping targets. And on the right, we can see our second method with our chain of association types and what databases we use for each relation type. We also try to prolong those chains with our last 2 examples on side effects and on cell lines, where we extracted relations of 2 separate open available databases. What did candidates look like in general? We had chemical elements, we had chemical compounds, which was the most observed type. example, yeah, of course, temozolomid, what is the main indication drug for the treatment of glioblastoma, and also bevacizumab, also known as Avastin. Then we had, for most interesting experimental vaccines, like the ICT-107, which were most interesting for us because they are often not available in databases, standardized databases. And we had adrenal cortex hormones and also various therapeutics which are more specific. For example, TT fields, which is an electronic device, also used for the treatment for glioblastoma. How did we evaluate our candidates? First, because we try to measure our potential of the text data, we benchmarked all our candidates with DrugBank, which is the most prominent and gold standard database for disease drug indications. For glioblastoma, it had 320 indications in general, ordered into two categories, one for known or main indication and a separate one on drug trials. So all indications which DrugBank tells us are in clinical trial. For that, we try to categorize our candidates into three categories. One was known in DrugBank, when our candidate was also known as an indication of glioblastoma in DrugBank, it was absorbed into this category. We had unknown candidates, that was when the candidate was valid but not in DrugBank. And we had invalid. Invalid most likely meant that it were mostly too broad or general terms of medications, for example, acid or other, or in general bad results of Ortega or the results. Here we have some prime examples of our results, which we mostly generated before we try to concept match them into single entities. What's mostly interesting is that temozolomid, of course, is mostly on top of all lists for known in DrugBank. And for our unknown candidates category, we have some very interesting candidates, like I talked about the ICT-107, which we, after short research, found was in just recent clinical studies indicated for glioblastoma. Yes. Important to see here is that in our second method, we saw with the variation by using gene variants and also of drug and cell lines, very bad results in general, which mostly indicates there aren't very that much gene variants or cell lines documented in those clinical text data. Some conclusion in general. What's the biggest point? We saw that the analysis of unstructured text data can enable a more comprehensive overview of potential repurposing candidates by providing up-to-date or lost knowledge. Overlapping genes as markers show the most reliable potential candidates for repositioning through our methods and use case. And that rarely any gene variants or cell lines were documented in our selected text data. So only symptoms, side effects, or genes seem suitable as possible embeddings for future analysis of text data. What were the main limitations? Of course, in the biomedical field, there is an abundance of synonyms. Why we had to use concept matching in general. We have either for a drug, we had a brand name, a compound name, or an experimental classification. Yeah, for that, it was necessary to use that controlled vocabulary. And we had limitations by the available text structure of open available text data. Those clinical trials which were in the database were mostly structured as keyword-like notes on clinical and trial reports, which also affected the performance of the models because most biometrical and year models are trained on full text data. For example, on medical corpora like Genia or Kraft. And yeah, with our approach, we just used a vague association approach. So most associations weren't really analyzed on their exact semantic connections, such as their possible causalities or negative relationships. And yeah, poor performance in recall and precision of size spacing in comparison to other biomedical available NER models like Stanza or Spark NLP. For this, I have to say, because it was started as a master thesis, this was mostly limited to my available computing power. And so, further research. Of course, we want to use text data also for research in the future. So we're gonna use better specialized NER models and also try to use transfer learning on them. Probably through Spark NLP. And yeah, we're gonna also use clinical NER models which can extract, for example, problems or tests or dosages in clinical text. And we're gonna focus also on more full text data from PubMed Central, which is available to researchers. So our scope will not be also limited by our selection of data, because we used a term like neuroectodermal tumors for glioblastoma. We were near that disease semantic-wise, so it's often talked about soft repurposing. But if we're gonna search for repurposing candidates in other text data, studies on other diseases, not cancer related, it's often talked about hard repurposing, and that is mostly our goal in the future, to find out repurposing candidates. And we try to more utilize knowledge graphs to contextualize our extracted relations and associations and identify contradictions in entity relations also. Thank you very much for your attention. I will be at the Swiss Data Science Conference in Zurich next week, where I will present additional parts and different aspects of this research. Thank you for your time.
Thank you very much. Maybe I can ask a question to start with. I'm really not familiar with the field. So now you have, it seems like you extracted large lists of candidates, like thousands of candidates. How does the process continue now?
Yeah, of course, we counted each occurrence and we ranked them accordingly. What was like the main thing to do. But of course, we're gonna talk with our experts, with our partners, to get more insight into why those candidates, those top candidates, which are potential unknown candidates, were that high on that list.
Okay, so you will, like, for finding actual good candidates, you will collaborate with experts.
Yeah, of course. We can't do that with data science because, and we don't have the resources to like do clinical tests by ourselves, so we are.
Thank you. Any questions?
Thank you for an interesting presentation. I have a question. Have you seen there's a lot of work in state of the art related to drug target interaction. It is to detect the interaction between the drug and the target, and then to classify this kind of interaction.
Yeah, you're talking about gene expression data, probably, like how much does a gene get regulated by a drug?
It's small protein.
Yeah, small protein. And mostly that data is mostly in databases, biomedical databases. So it's not really was that much of a focus on analyzing text data. But for future research, we of course will also look at those types of data more.
Yeah, in fact, you can, there in the abstracts of the articles, kind of introduction in one sentence, you see that there is two occurrences of one is a drug and the other is a protein, and there is kind of relationship that is 4 or 5 relationships according to the specialist.
Okay, good to know. Yeah, that would be an interesting part. Yeah, but then we needed to use more background on what how to say how that protein should change based on glioblastoma, what we eat.
Exactly. Yeah.
Exactly. And mostly those kind of topics are a little black box in the biomedical field because sometimes they don't really know or can measure everything which was like had an effect on which proteins. So, but very good question, yeah. Thank you.
Any more questions? Can you elaborate on what you have then done concretely? I mean, you're talking about repurposing, which means you have a drug which is always in the market or clinical studies going on, and it's been done for a specific disease. And now you're talking about repurposing, so I didn't get this from the way I missed it.
We try to like get a list of possible candidates which we can rank by machine learning techniques to get more prioritization for clinical tests for repurposing. With the whole data science aspect, we can't really say, okay, just try this drug, because our main goal is just to provide prioritization of possible candidates for an indication.
But for known disease,
For known disease, yeah, of course.
because
Yeah.
when I hear about repurposing, it knows for me that it's for a different disease for whom it is originally being put into clinical study or market. So that was why I was a little bit confused now. So you are sticking still to the original disease which it was planned or the clinical study was planned, or is it really that they've gone into other clinical studies where it originally was not planned? Like it's a different, they targeted like retinal diseases, for the eye, but now they figured out, oh, these also looking for heart.
Yeah.
Okay. So is it this type of thing, or you were looking for the retinal diseases and say, hey, who else has also done something on it?
No, it was the first thing you described, yeah. That's why we took like a soft repurposing approach with the broad term of glioblastoma, neuroectodermal tumors, which also includes glioblastoma or cancer of the main tissue.
Okay.
Yeah.
Stick to the area.
Yeah, but yeah, the goal of course is to go out of that reach and go into the direction of heart repurposing, which is like, oh, there's a study about heart disease, oh, that drug has an indication possibly for glioblastoma.